So we are now welcoming Dr. Samantha Paoletti from CSEM Neuchâtel. Thank you so much for being with us. We're looking forward to your speech.
Good afternoon, everybody. It is my pleasure to present you what we are doing. It is my pleasure to present you what we are doing at CSM in regards to the development of new innovative technologies that can change the way we treat and we support cancer patients. Few words about CSM. So we are a Swiss innovation center, and we position ourselves in between fundamental research that is done greatly at the university and industrial and companies. So we support companies in regards to the innovation valley of death. We are 630 employees. We are 40 years old. We have above 100 million turnover, and we have a patent family that will access at the industrial sector. And I will try to give you an understanding of different technologies that are developed actually by the 630 employees that can really revolutionize the way we address healthcare, and with a special focus at cancer. So that is a number of products that recently reached the market with a strong focus on life science, going from precision liquid handling to on-body monitoring. And in many of these product or application, actually, there was behind a new way or to reduce the healthcare costs, or to reduce the drug production, or to speed up the time we need to new drug to the market. And looking at maybe more in details the example of cancer. So we heard from the first presentation that one of 3 persons in the life, they will be diagnosed with cancer. We heard that there is a great achievement in regards to the way we can treat patients, but also with the with increasing number of patients that will be diagnosed with cancer, we need definitely new treatment. And that was a lot of work in the past years, going from more conventional treatment to cell therapy, and a lot of work that can still be done. And the second part is that we need to have better tools for diagnosis, for make prevention, and also to support patients with cancer to constantly monitor themselves without the need of visiting the medical doctor. And ourselves, we are developing technology that somehow can support these overall pictures. When we look at the topic of new treatment, if we look at the overall cost of drug development, that is a quite costly investment that should be done. And one of the areas that is quite costly, where there is a high failure, is preclinical testing. So there are still a lot of drugs that in the preclinical testing, they look positive, and then they enter clinical testing, and that is a moment in which they fail. And that is because in preclinical testing, we have very few tests that are really giving us an information about the efficacy and the potential toxicity of drug. So far, we were using animal models. It's a very valid information that are provided, but of course, we are not, as human, we are not as similar as mouse. We use cell culture in 2D. They were also informative, but actually in our body, we are done as a 3D. And that is a potential of using 3D organoids. So organoids are 3D tiny cell culture. So we build in the 3D how it is the tumor. And then we can use it for drug screening and also for disease modeling. So it is a lot of work that happens in the past years. It was a research activities for many years. And since the past 20 years, so 2 decades more or less, pharma domain has a strong interest into that, because they realize that it could be extremely informative tool in the preclinical testing. Now, if we want to use it in the pharma domain, we should be as close as possible to the human physiology, and we should be able to standardize and increase the throughput of the technology. So in order to be used by pharma, we should have a high throughput of production of the organoids. We should be able to fully automatize the production and the handling, and they should be very standardized. So if I have one organoid per well plate, I should make sure that they are all equal, because then I will expose them to different drugs, and I want to have my results based on the exposition of the different drugs and not of the different biologies that is happening behind. So standardization, automation, and increase of the throughput, that is the work that we do at CSM. We don't focus so much on the biology because that is greatly done already by many academic partners and also by the industrial domain, but it's really the technology that are around that will allow this technology to become a preclinical high throughput screening. So tumor organoid is one part. You see here a little bit the size. There are different ways to create tumor organoids. You can start with different starting material, and that it depends a lot on the biological question you want to ask the chip. So which question I want to ask is usually making people choosing a different type of tumor organoid. But we heard also in the first talk that actually our body is heavily impacted by the fluctuation of blood and immunocells. And so far that was a hard part. So we could build in vitro organoids in a static way, but we didn't have a way to making immunocells circulating in a standardized way. So vascularization, as I said, is part of our body. All our organs are constantly perfused by blood and constantly perfused by immunocells. And it is a unidirectional fluctuation or flow, and it is a continuous flow. And that is what we did build in vitro. So in organoid chip, also all microphysiological system, the majority of the time people, they use a well plate. That is a super well-known labware that biologists, they like and they want to use. Is also compatible with automation. And usually the knowledge or the biological IP stay in the well plate. And then by working in the past years, we realized that somehow there was one part that was never touched that could add additional features. So if we think, sorry, I go back shortly. So people, they use a well plate, and then on top of the well plate, usually you have a lid, and the lid is mainly used only to keep sterility. There is not any other function on top of the well plate given to the lid. And that's what we thought. Was like, okay, can we give functionality to the lid? And that was one of the first projects. So we brought microfluidic inside the lid. So there is a part in which we can insert media, a part in which we can remove media. And then by doing microfluidic in the lid, we can address all the different well, and that media was mainly used for media exchange. So you can imagine that you have your cell culture, you have the smart lid on top, and you can exchange the media without the need of opening the lid. So that is a big advantage because we don't need to take out the cell culture potentially from the incubator, and it could be fully automatized. So that is already transferred in different industrial contexts, and it is already used by some companies. We brought also additional features into the plate. We were able to bring perfusion inside the plate to help support again the way in which our body is functioning. And the last one that I will show you more in detail later is a vascularization lead. That is probably the most sophisticated lead that we did so far. And with that, we can build a unidirectionally continuous flow inside the well plate where tumor organoid are built. So that is how does it look, the technology. You can see that we can address each line independently. So it is a modular way that we can have it. In this way, we can decide if they are all needed or how we can address potentially the line in a different way. Below that, there is the ident plate, so the ident TX40 or also the TX3, so the one from IMI Biotech. And that for us, it is quite independent. So we can address different well plate independently from, so maybe we will have to change the position of how the things are done, but somehow we try to be as much as independent from the design of the well plate underneath. And that are the results. So inside the plate, that is a collaboration we had with Roche. Inside the plate, they were able to build vascularized blood vessels. That is what you see in dark blue. We were able to have patients-derived tumors. That is what you see in green. And we had circulating immunocells. So the cells were circulating for several days into the culture. And that is quite an amazing feature because one of the challenges with microfluidic and cells and organoid is that the single cells need to fluctuate. They are quite fragile. The tumor organoids are 3D cell culture, so should be treated also in a gentle way. And the same is for the vascularized blood vessels. So this technology allowed to bring more functionality in vitro models and preclinical testing, and to be much closer to human when we try to test, first, when we want to try to understand the disease, and second, when we want to try to test a new type of treatment and of drug. Changing completely the topic, so the first part I wanted to say that there is a big potential for new technology and innovation in the preclinical testing. The second part is either prevention or either is how we monitor patients at home. And I think in the case of cancer, definitely it is more how we can keep patients at home. And here at CSM, we work since many years on wearable solution. That is, I think, is something we are all extremely proud. It is a startup. His name is Actia, and is measuring blood pressure in a continuous manner. It's not a gadget. It's not a consumer health gadget. It is a medical device. It's a CE mark medical device, and it is used a lot for the hypertension management. Similar tools, adding additional functionality, are used to constantly monitor patients at home without the need of them of visiting the hospital very often, because that is, of course, increasing drastically the healthcare costs that we have at the moment. And that is an activity running since very long. We have different physical parameters that we can monitor. On top of that, we started to bring a biosensor. And that is one of the solutions. Is not cancer related. We have actually two projects that are more related to cancer, but they are in industrial medical, so I cannot disclose the details. But there is a possibility in the similar form factor, so extremely miniaturized, to monitor also different parameters. And here we work with electrochemical sensor. So inside there is a screen printed sensor. We have an electronic readout that was done for saliva monitoring in the mouth. But similar to this, we have many technology in the context of microneedle. And with the microneedle, we can constantly monitor some parameters up to 30 days, similar as glucose. And now it is a field that before was really only used for diabetes management, and now is used more and more for many chronic conditions, and also conditions where you need to monitor continuously, potentially also the potential effect of a drug. And that is maybe just very quickly, we're going to have two workshops very soon. Is maybe the one related to, could be related to cancer is the second one, the single cell omics workshop. So if you are interested, we would be happy to share more information about that, and happy to take your question now.
Thank you so much, Dr. Paoletti, for this inspiring speech. It's very impressive how you accelerated the transition between academia and industry. And you have such an impressive portfolio of products you are involved in. So there's one question from Sarai. Thank you so much, Sarai, for your question. So just to clarify, the tumor tissue is derived from a single individual patient, individualized organoids. Because if so, this is amazing for personalized medicine.
Yeah, so that would be the big dream of everybody, that before, so I discover I have cancer, I would not be, of course, very happy. But before I start any treatment, they take a little bit of my tumor cancer stem cells, potentially. They build my organoid, they expose this to different treatment, and I understand which is the treatment I'm going to react. So that is going to give me the chance to be treated immediately with the right treatment. We are still not there. I think it still takes some time. First, because it's still a technology that is not entered the clinics. It's still a technology that is mainly in the domain of pharma. So we need to still make the transition also from pharma to the clinics. At the moment, it is mainly used as a clinical or as a research tool, or in the pharma domain for drug screening. But definitely the big goal would be definitely what Sarah mentioned, that I take my cells, I put it in the chip, and I understand which is the right treatment for myself.
That's impressive. So Antonia is asking, how user-friendly are those wearables in mouth?
So we had just a colleague that entered because the company, the iTooth, they do now the clinical validation, and a couple of colleagues that were keen in participating in that. they manage. So it's not like a wearable that is not a bracelet that you take it for days, day and night. Usually it is something that is used for a couple of days, and I think it's fine. What we discovered doing this project is that there is a space in the mouth that actually we produce, all of us, a lot of saliva. Not all the saliva is the same. So if I change the position in the mouth, I might get slightly different information because there are different glands that produce saliva. So standardization of sample collection and preparation is something extremely important in point of care. And the other thing is that there are different mouth size. So people with the bigger mouth at the moment, I think it's a little bit more convenient if it is a smaller size. But definitely there is still a potential to miniaturize even more this kind of technology.
Thank you so much. So Sarai is asking, how are the devices designed for continuously monitoring powered? If worn, e.g. in the mouth, are there electrical considerations?
Yeah, so we work a lot. We have a strong knowledge. So the one in the mouth, but all the wearable solution at CSM, we have strong knowledge on the ultra low power. So we know how to either try to harvest energy from the environment. That is not the case of the mouth, either really working at ultra low power. So, and that, of course, when you are in the mouth or in the body, there is a strong focus we have on biocompatibility. So to make sure that somehow we are not generating damage to the, so that is a big, so miniaturization, ultra low power, and also biocompatibility is a strong focus that we are having in all the development.
Thank you so much.